got it, okay. A very good morning to everyone present. Myself, Dr. Shruti Attawar from Department of Conservative Dentistry and Endodontics, welcome you all to the lecture series on Sustainable Developmental Goals. Today's lecture is on climate change and health, which is organized by A.B. Shetty Memorial Institute of Dental Sciences in association with Staff Developmental Cell, Nitte, deemed to be university. It's my privilege to welcome today's dignitaries, Professor Dr. U.S. Krishnanayak Sir, our beloved Dean, the resource person for the day, Dr. Idyakarna Sagar Sir, who is the advisor of research and patents, Professor Dr. Smita Hegde Ma'am, Deputy Director, NUC Sir. I would like to give a few insights regarding climate action, that is the SDG 13. Climate change is a real and undeniable threat to our entire civilization. The effects are already being visible and will be catastrophic unless we act now. Through thorough education, innovation, and adherence to our climate commitments, we can make necessary changes to protect our planet. These changes also provide a huge opportunities to modernize our infrastructures, which provide prosperities around the globe. Now, I would request our beloved Dean, Professor Dr. U.S. Krishnanayak Sir, to give his opening remarks.
Thank you very much, Dr. Shruti. Most respected and revered Professor Dr. Idyakarna Sagar Sir, the Advisor, Research and Patents of Nitte DU, other heads of institutions who have joined for this lecture series on Sustainable Development Goals, the coordinator of today's program, my own colleague, Professor Dr. Shruti. I, on behalf of our beloved Pro Vice Chancellor, first of all, would like to apologize for him not being able to make it today. And in fact, he was supposed to be the one who was going to give the opening remarks, but due to unavoidable circumstances, in fact, a core committee meeting was convened at the last minute. So Sir's presence at that meeting was very much essential. So that is why he could not join in. However, he has asked me to convey his apologies to the audience and each one of you for not being able to join in after having committed for this. All the other senior officers of the university, my colleagues, staff members, postgraduate students, my colleagues from other institutions, and friends. It is indeed my privilege and pleasure to first of all welcome each one of you to this lecture series on Sustainable Development Goals. Today, we are going to talk about climate change and health, and the resource person is our own, I would say, most respected Dr. Idyakarna Sagar, who is going to talk to us about climate change and health. Now, this is a burning issue of today. Already, you can see the climate change all around us. The rains are still continuing, the global warming, etc. We can all see it. So unless we really try to arrest this trend, we are really going in for something really catastrophic, if I may use the word. The Sustainable Development Goals are a collection of 17 interlinked global goals designed to be a blueprint to achieve a better and more sustainable future for all of us. These goals were set up by the United Nations in the year 2015 and are intended to be achieved by the year 2030. With all our combined efforts, I'm sure we will be able to achieve this across the world. And we have no choice at this. We have to definitely make sure we achieve it at the earliest, latest by 2030. The sustainable goals were adopted as a universal call to ensure that all people in the world enjoy peace, prosperity, and we protect our planet. Some of the important goals are all very important and important to life. And today's topic is on SD, that is Sustainable Goal 13, that is climate change and health. This, of course, all of you would agree is a very important topic because we need to save our ecosystem and that we can preserve natural resources for our future generations. Climate change is in simple terms, describes the changes in average condition, such as temperature and rainfall in a given area. We can already experience the warming, the rising sea levels, the shrinking mountain glaciers, etc. These changes can harm food production and, of course, human health in a large way. Hence, as a university, it is our duty to generate and translate and disseminate knowledge relevant to achieving these goals. Measures to attain these goals are nothing new to our university. Some of the activities that we have been practicing over the years are the very design of our campus is such that we have got very large inner spaces that provide light to the indoor plants, which provide a healthy oxygen, rich air, and reduce the carbon dioxide level in the atmosphere. We have a terrace garden which have got about 50 fruit trees. Plant a tree on your birthday, celebration of World Environmental Day, and most recently, just a couple of days back, we commemorated Farmers' Day by planting trees near our boys' hostel. We have been ranked, as all of you are aware, 253rd among the world's most sustainable universities and given by the Metric University World University Ranking. Now, without taking much more of your time, I once again thank Professor Dr. Idyakarna Sagar for accepting our invitation to be here and deliver this lecture on climate change and health. Before I go ahead and request Dr. Idyakarna Sagar to start with his lecture, I would request Dr. Shruti to kindly introduce, though he is our own faculty and somebody who does not require introduction to most of us for the benefit of some of the students, UG students who may not be very familiar with Sir. I would request Shruti to kindly introduce Sir to the audience. Thank you very much. And have a nice day ahead.
Thank you, sir, for your kind words and support. Now, I would like to introduce the resource person of the day. Dr. Idyakarna Sagar obtained his master's and PhD degrees in microbiology from Mangalore University. He carried out postdoctoral research at University of Maryland, USA, University of Sendai, Japan, Natural Resource Institute, UK, and University of Würzburg, Germany. He joined University of Agricultural Sciences, College of Fisheries, Mangalore as assistant professor, rose to become associate professor in 1982, professor in 1992, head of Division of Fisheries Science 2003. When Karnataka Veterinary Animal and Fisheries Sciences University was carved out of UAS, he became the first director of research. Indian Council of Agricultural Research awarded him National Professorship in 2005. During 1995 to 2005, he served as drafting member of Joint FAO-WHO expert meeting on microbiological risk assessment. He then moved to Food and Agricultural Organization of United Nations and was based in Rome as senior fishery officer, where he led a team on fish safety and quality at FAO headquarters in Rome. He played a key role in several FAO-WHO food safety risk assessment, which led to the development of international food standard at Codex Alimentarius Commission. He represented FAO in meetings of Kodak's Committee on Food Hygiene and Kodak's Committee on Fish and Fishery Products. He was lead technical officer for several food safety products in Asia, Europe, Africa, South Pacific, Caribbean islands, and Latin America. He played a pivotal role in development of WHO 5 keys for safer aquaculture products. Following his retirement from FAO, he has been working as international consultant to organizations like FAO, WHO, Asian Developmental Banks. He is also a senior director at Nitte University since 2017. Dr. Idyakarna Sagar has guided 30 PhD students and 45 master degree students. His work on developing and commercializing molecular diagnostic technique has been recognized by the Ministry of Science and Technology, Government of India, with the Biotechnology Product and Process Development Award in 2003. He has over 220 publications in referred international journals and a H index of 40. He has 15 book chapters and 1 book published by international publisher. He has 6 patents and 3 commercialized technologies. He has 5 bacterial genomes, 2 viral genome, and over 4,000 nucleotide sequence in his gene bank. He has been elected Fellow of National Academy of Agricultural Science, Indian Academy of Microbiology. He has served as member editorial board in several international journals and is currently academic editor of PLOS 1, associate editor of Journal of World Aquaculture Society. He chairs the scientific panel of Food Safety and Standards Authority of India and Antibiotic Residues. Over to you, sir.
Sir, kindly pardon me. My apologies. I'll be failing in my duty if I do not congratulate Dr. Shruti for planning this lecture so meticulously and executing it to perfection. Shruti, kindly accept my hearty congratulations on the same, and I wish you all the very best. I'm very sorry for interrupting, sir. Thank you.
Thank you, sir.
Thank you. Thank you very much, respected Dean, Dr. Krishna Nayak, and heads of institutions and all the university officers, faculty, and students. It's indeed a great pleasure for me to be part of this program, and I'm aware that the A.B. Shetty Memorial Institute of Dental Sciences have been doing a lot of work in this area of SDGs by organizing so many lectures by different resource persons highlighting the importance of SDG. And the Dean has already explained the various aspects related to SDGs and how it is important in our life, and how Nitte is what Nitte is doing to highlight these and be one of the sustainable campuses. So I will start my talk. Are you able to see my screen?
Yes, sir.
Okay. Okay, so the topic of today's talk is climate change and health. Of course, Nitte being a health university, I thought this will be an interesting topic. And we are all aware of the impact of climate change on human health. And human health and environment is so closely related that today people talk only in terms of one health, where human health, animal health, and environmental health, including plant health, are closely interlinked, and we cannot consider any of these in isolation. So we need to consider human health, as well as the environmental health together. Now, what is the impact of climate change on health? Now, WHO estimates that between 2030 to 2050, climate change is expected to cause additional 250,000 deaths every year from malnutrition, malaria, diarrhea, and heat stress. So as you can see, malnutrition coming mainly through the impact of climate change on food systems, food productions, and food availability and supply. Malaria due to the change in the spread of vectors. diarrhea due to impact on water availability, heat stress. So these are some of the major impacts on health. And the direct cost to health, excluding indirect costs related to agriculture, water, sanitation, etc. So only the direct cost is estimated to be 2 to 4 billion US dollars per year. And developing countries like ours with weak health infrastructure are the most vulnerable. So here you can see a summary of the impact of climate change on health and how the vulnerability is affected and what are the pathways of exposure and what are the health systems. Now, exposure pathways include extreme weather events. We have been experiencing many of them: untimely heavy rains, floods in different parts, landslides. So these weather events have been impacting the ecology and also health. And heat stress, temperatures going above 40 to 50 in some parts. Air quality. Now, because when people go to Delhi, they always complain about air quality there. And then water quality and quantity, availability of water is a very scarce resource. Food security and safety, and vector distribution and ecology. Now, these exposure pathways are interacting with various vulnerability factors like demographic factors, geographic factors, biological factors and health status, socio-political conditions, and socio-economic factors. Because in India, we always say we have a demographic advantage, we have a young population, but we are also susceptible in some of the areas where we have vulnerability, which is high to some of these exposure pathways. And how these can be counteracted with health systems. We need leadership and governance, health workforce, health information systems, essential medical products and technologies, service delivery and financing. So the climate sensitive health risks are again summarized in the lower portion of the slide. You have injury and mortality from extreme weather events. We saw this in Uttarakhand in February, in many parts in Kerala, floods, Coorg, so many landslides and loss of homes occurred. Then heat-related illness, respiratory illness, and waterborne diseases, zoonosis, because now we are in the midst of a COVID pandemic, and we don't have evidence to show that COVID is a result of climate change, but there are other zoonotic diseases, the Nipah virus and so many other viral diseases, vector-borne diseases, which are affecting humans due to the climate change. So the vector-borne diseases are also covered. Then malnutrition and foodborne diseases due to impact on agriculture and other non-communicable diseases, and mental and psychological health. So the health systems and facilities need to overcome these outcomes. So if you look at what are the impact of climate change? So you have rising temperatures, you have more extreme weather, we have rising sea levels and increasing carbon dioxide levels, and the resultant changes in vector ecology, increasing allergens, water quality impacts, water and supply impact, environmental degradation, extreme heat. So all these, for example, extreme heat will cause heat-related illness and death and cardiovascular failure, severe weather injuries, fatalities, mental health impact, air pollution causing asthma and cardiovascular disease. This is already very prevalent in several parts of India. And changes in vector ecology, malaria, dengue, encephalitis, hantavirus. So there are so many vector-borne diseases. Because in India, we face malaria, dengue, chikungunya. These are very common in India, and they are occurring during different periods of the year, not only seasonal. Increasing allergens, respiratory allergies, asthma, waterborne diseases like cholera, cryptosporidiosis, campylobacter, leptospirosis, and harmful algal blooms. I'll be talking a little bit later on that. And malnutrition, diarrheal diseases. And this is all leading to, say, forced migration, civil conflicts, mental health impact. So it's a very complex issue. And there are a number of different impacts of climate change on health. And melting of glaciers is one of the very important aspects which is affecting many of the ecological changes that we are seeing. And it is estimated that between 2000 to 2019, the melting of glaciers has led to 21% sea level rise. Because in sea level, even if it is a 1 inch rise, it will be catastrophic. So this glacier melting has already caused, because oceans cover 70% of the Earth's surface. So it's such a huge water mass, and a level of rise in even 1 inch could be catastrophic. So all this melting of glaciers have already caused sea level rise, and another 15 to 25 centimeters sea level rise is expected by 2050. So these are some of the images of the same location taken, say, in February 1913 and January 2018. So you can see the impact. And in India, we already experienced this, Algal, the flash floods in February, which led to loss of several lives and so many damages. So you can see the river, and after the flash floods, how the total river is muddy, and the whole water system is affected. So I will try to focus on a couple of aspects, particularly the harmful algal blooms, which is an important aspect, particularly in the coastal regions, since we are located in the coast, and we are experiencing these harmful algal blooms. So these are blooms of algae in the oceans, particularly in the coastal region, and how it is affecting the fish and the fish consumers, and lead to toxicities and health impacts. So this summarizes the events that have been occurring since, say, 1908 to 2017. As you can see, between 1998 to 2017, there have been a massive increase in the incidence of these kind of algal blooms which are taking place in the oceans. And you can see what are the different species of algae that are causing these blooms. And so this illustrates some of the effects, like injury to fish and other aquafauna, and shellfish poisoning, Sigmoidal fish poisoning. I'll be talking a little bit about these little later. And also the fish kills that are caused, and what are the different types of algae which have been causing this kind of blooms along the Indian coast. So here you can see how harmful algal blooms along the coast are impacted by increase in sea surface temperature, increased nutrient flow due to heavy rains, storms and hurricanes. So the impact of these would be on fish. So it reduces the availability of fish, affecting nutrition. So fish kills, fish become toxic, as we will see later, and events like Sigmoidal fish poisoning, which affected large number of people near Mangalore some time ago. So the various environmental factors like increase in temperature, increase in stratification, increase in ocean acidification, cultural eutrophication, and grazing affect different types of algae in the oceans, and how this is impacting the ecology and fish availability and the safety of fish. Now this shows the impact algal blooms which have been occurring in the Indian Ocean region, not only in the Indian coast, but also in the Middle East. For example, the Arabian Sea, UAE coast also has been experiencing some of these algal blooms. And this illustrates the incidence of a type of shellfish poisoning called the paralytic shellfish poisoning. So if you see in 1970, there were only a few dots along the American coast. So there was nothing in Asia, there was nothing in India. So most of the parts did not experience this shellfish poisoning, paralytic shellfish poisoning. But in India, we reported for the first time an outbreak in Arikadi, which is close to this place, where some people died and several people were hospitalized after consuming clams, which were toxic due to the presence of these toxin. And now you see in 2015, almost the whole globe is affected by this paralytic shellfish poisoning. So this impact, you can see the impact on fish health, the human health, through this fish, which is a very important resource. So when we did this study in Arikadi, and we were looking at the consumers, mostly these clams are consumed by poor people, and clams are a most important protein source for this population. So if clams are not available, then their nutrition is impacted, and so the sole source of protein which is available to them will also be not available. So this has been occurring throughout the globe. So now it has impacted almost all countries. So this was the study we published in 1990. Of course, the outbreak occurred in 1984 when we initially did some mouse bioassay and demonstrated that it is the shellfish toxin. And then finally, we could characterize the toxin profile, because it's a very complex toxin which has to be characterized only by HPLC. So it was not common to find HPLC in laboratories those years. So we did not have HPLC at that time. Today, of course, HPLC is very common in laboratories, and you see even undergraduate students may be using HPLC. But at that time, it was a very precious instrument. And to characterize them, of course, you needed some special type of system where you have to derivatize these toxins after separating them through a column, and then characterize. Because there are so many different moieties that are being involved in this shellfish toxin. It is called saxitoxin, and there are several analogs like guanetoxin, neosaxitoxin, and decarbamyl toxins, and decarbamyl neotoxins. So there are more than 16 chemical moieties, and these vary in their biological activity. So just detecting a toxin is not sufficient, but you need to determine whether it is really causing biological activity, what is safe, what is not safe to consume needs to be determined. Now, these have been summarized. Of course, this came out after a joint FAO. So we did a compilation of all the risks, biotoxin risks associated with bivalve mollusks. And this was presented to the Codex Alimentarius Commission, which was working on the bivalve standards. So then the Codex could come up with these standards. So there are standards for saxitoxin. So the minimum, the level, the tolerable level is less than 0.8 milligram of saxitoxin equivalent. So here all the standards are in terms of one representative chemical moiety. So then we need to also, if there are so many different groups of toxins, how do you convert these? So that there are issues related to that. But the standards relate to one reference molecule, which is representing. So there are saxitoxins cause paralytic shellfish poisoning, okadaic acid toxins cause diuretic shellfish poisoning. So this was also detected in the coast of Dakshin Kannada in the shellfish. It causes diarrhea, so it's mild illness, but it may not be differentiated from say other foodborne illnesses caused by bacteria, but it is a mild one, fortunately. But these toxins are also causing health impacts. Domoic acid, there are other toxins which are having neurotoxic effect, amnesic shellfish poisoning, and other toxins. So then how to convert these toxin moieties into the reference toxin? So this was again brought out through an expert meeting in FAO, where a group of experts came together and looked at the toxicity of different chemical fractions and determined how we can convert these into the toxin, reference toxin levels. So this is very important for managing shellfish safety. In many parts of the world where bivalve consumption is regular, there is a regular monitoring program. So people have to look at the food safety authorities have to look at these toxin levels and either permit the shellfish harvesting or not, depending on the levels that are present in these shellfish. So another aspect I'm going to talk about is on Sigmoidal fish poisoning. Now this is again another important aspect, because this is caused by consumption of fish from coral reef area. And earlier it was thought that it is not important for countries like India because we have very little reef fish that are coming to our coast, so people do not consume this. So this poisoning is associated with the type of algae called Vibrio species. Now, over 400 fish species can be carriers of this toxin. But the common ones are red snapper. Because this is commonly, you can find them in the fish market in Mangalore. Red snappers, reef cod, groupers. Groupers are also common in the fish market in Mangalore. Barracuda, kingfish. Kingfish is also common here. Trevally, parrotfish, wrasse, giant moray. So different fish can carry this toxin. So if the toxic algae are present in the water, then these algae are ingested by these fish and the toxin accumulates in the body of the fish. So the fish will look absolutely nice. It may be looking fresh, nice fish, good bait, but then it may be carrying the toxin. So the problem with these shellfish toxicity, both the paralytic shellfish toxins and the Sigmoidal poisoning, is you cannot make out when you look at fish. So when you go to the market and see if, the fish, whether it's good enough for buying, you may think, oh, this is a nice looking fish, so it must be very good, but it may be carrying the toxin. So we need to be careful about some of these kind of events that have happened and how it is impacting health, and this is related to the climate change that is taking place. Now this Sigmoidal fish poisoning is caused by a heat stable lipophilic toxin, and it's highly potent, and ingestion of as little as 0.1 microgram of toxin can cause illness in a human adult. So it's very potent. And the saxitoxin, about which I talked about, it is even more potent. And in fact, in the 60s and 70s, some of the countries have extracted toxins from tons and tons of shellfish for biowarfare. So this is a molecule which is which has potential for even biowarfare. So in fact, when I was to go to US for a conference on this shellfish poisoning, when for a US visa you have to appear for an interview. So the interviewer asked, what is the conference I'm attending? When I told him it is to deal with the shellfish, then he got a little bit scared. And he said, okay, we will have to confirm. So your visa issue will take some time. So it took nearly a month for me to get the visa because they had to get back to the conference organizers and check what is the activity I am doing in India. So this has also impact from other angles like the biowarfare, so very potent toxins. So the symptoms in the case of toxin usually start between 1 to 48 hours of eating fish. And the symptoms include both gastrointestinal and neurological as well as cardiological symptoms. Now, Sigmoidal fish poisoning, again, you can see the region where it has been reported. From India, there were no reports. So in some parts of the world, it has been occurring, and it has been reported to be mainly confined to certain parts of the world where you have these reef fishes, and these algae are associated with these reef areas. But then this happened, so there were patients admitted to the K. Hegde hospital. I think about 100 people were admitted here, and I think more than 200 people became sick. Now, this is a very interesting incident because most of the people who ate this fish were from a fish processing factory. And this fishing processing factory was located in Ullal, and they were processing the fish, removing the head, and then the body of the fish was going for export. And export, there was no problem because the body does not contain the toxin. So most of the toxin is present in the head. So the head, which was retained in the processing factory, was consumed by the factory workers. and then they became sick. So most of the people who became affected were these factory workers. So the body, so we need to know which part of the fish contains this toxin. So it's very important from public health point of view, how to manage this shellfish toxicity. So it's only through issuing advisories. So the clinical presentation, it was mainly showing, people were showing typical symptoms, and the fish involved was red snapper. So what we did was, we did a mouse bioassay, and then we also collaborated with a laboratory in US, which is doing a receptor binding assay, because Sigmoidal assays are not common in many parts of the world, because not a toxicity that occurs in several different parts of the world. So only there are certain regions which have expertise in this. Of course, US has expertise, Australia has expertise, because there are a number of Pacific areas where this kind of toxicities can occur. So this type of assays are done only in limited parts of the world. So we collaborated with the laboratory which did this receptor binding assay, which showed that this is in fact a Sigmoidal toxin. And then this was confirmed through liquid chromatography and mass spectrometry, which was again done in collaboration with a laboratory in UK, which is a bival reference laboratory. And they have been working in different parts of the world looking at this type of toxicities. But one of the most critical issues here is that these toxins are vary. There are different types of toxins, as I will talk later, and you do not have a reference standard. So there is a major issue when it comes to determining the presence of toxin and determining the toxin levels. Now here, Gambier discus involved in Sigmoidal fish poisoning produces two types of toxins: water-soluble meatotoxins and lipid-soluble sigmotoxins. Now, if you do a mouse bioassay without knowing these different types of toxins that occur, then you may come to a wrong conclusion, because even these meatotoxins can kill mice, but they have no role in Sigmoidal fish poisoning. So if the mouse bioassay is positive, then you need to rule out the presence of this meatotoxin. And then you have a very extensive extraction and cleanup procedure before you can detect the presence of this sigmotoxin in fish. So now the geographical range of this Sigmoidal fish poisoning has been extending because of climate change. So in East Asia, Southeast Asia, and also now Indonesia and India. Because India, we had this experience, and then from other countries also now they are reporting. So Sigmoidal fish poisoning is being reported from geographical regions where it was not thought to be a problem before. So here you can see the impact of climate change. The geographical range of some of these toxic species presenting in the ocean is changing. So the range for this Sigmoidal fish poisoning was considered tropical and subtropical, between 35 degrees north and 35 degrees south. But now it is known to occur even in temperate regions of Japan. Even Japan experienced Sigmoidal fish poisoning. Southern Australia, New South Wales, where the temperatures are low, but still Sigmoidal fish poisoning occurred. And even in Europe, Canary Islands, Spain, Madeira, Portugal, Greece, Gulf Coast. So there are new geographical regions from where this Sigmoidal fish poisoning is being reported. So even incidence in endemic regions is increasing. Like for example, in the French West Indies, incidence during 2013 to 16 is 5 times higher than the incidence during 1996 to 2006. So not only the geographical range is expanding, but the frequency in endemic areas is also increasing. And this is attributed to the disturbance caused in the reefs, and which is attributed to the cyclones. So how climate change is impacting ecology, and then the food safety. So over 400 species, fish species may be involved, both herbivorous and carnivorous species. So you cannot avoid fish based on whether they are herbivorous or carnivorous. And identifying fish species that can carry toxins is important. So people need to be aware. So in regions, for example, in the Pacific Islands and also in the Caribbean region where it is common, the local people are aware. So they know what fish to eat at what time. So they can avoid because of traditional knowledge. But in new areas, people are not aware. That's how it happened in Bangalore. People didn't know that this red snapper carried this Sigmoidal fish poisoning. And so there is significant variation in the toxin content between individual fish of a species and between geographical areas. And interestingly, even new fish species are being identified as carriers. For example, sharks in the Indian Ocean. So they can also be carriers of the Sigmoidal fish toxin. So this toxin is again a very complex chemical molecule. It's a polyether toxin. And three major groups have been identified. One is the Pacific type, so what is occurring in the Pacific Ocean. And another one is the Caribbean type, the sea type, and the Indian Ocean type. So we still do not know much about the Indian Ocean type because these are all now new areas which are occurring. And so far, there are these two families, that is Pacific Sigmoidal toxin. This again is grouped into type 1 and 2. And there are so many congeners of each group. Now, for example, PCTX1 to PCTX4, and again the Caribbean type CCTX1, CCTX2, and up to CTX4. So there are so many chemical variants of these toxins. So this is a real challenge for chemists to determine which toxin is really present. And again, as I mentioned, it is difficult to have a reference. You need, for all these chemical estimations, you need the reference toxin. So the reference toxin is still not available. So the Pacific Sigmoidal toxin is considered most potent, being tenfold more potent than Caribbean Sigmoidotoxin. So Indian Ocean ICTX variants are considered 60% potent compared to the Pacific type. So there are a number of technologies that can be used to detect. Of course, the first one is the mouse bioassay, which can be easily applied in many laboratories. And then there is a receptor binding assay using neuroblastoma, and also cytotoxicity assay and receptor binding assay. And then LC-MS. So you need liquid chromatography with tandem mass spectrometry to characterize this toxin. So the reference standard toxins are available only few laboratories. So it's not commercially available. And these laboratories have purified their own toxin from the fish. So they have their own home-derived reference toxin, but commercial reference material is still not available. And purified Indian Ocean toxins are not available. And acceptable levels of Sigmoidal toxin are not agreed internationally. So the US FDA is using a level of 0.1 parts per billion for Pacific Sigmoidotoxin and 0.1 parts per billion for the Caribbean toxin. So the level varies because the biological activity of these toxins vary. So you need information on the toxin, what type of toxin is prevalent and what is its biological activity to determine the level, acceptable level. So internationally still there are no codex standards for sigmotoxin. So particularly after these incidents in the tropical region in India, there was an FAO expert meeting which was organized. I'll mention about this a little later. So monitoring reef areas for the presence of Gambia discus species is done in some areas. There can be, but more than 100-fold variation in toxicity between species. So having information only on the presence of algae may not be sufficient to predict the incidence of toxicity in the fish. And identification of susceptible fish species would be important because then locally people know, but there can be again large variations in toxicity. And as we saw in the red snapper in Bangalore, even within a fish, the different parts of the body. So head contains most of the toxins and not the body, the muscle region. And avoidance of large fish. So the general advice given in areas which are susceptible for Sigmoidal fish poisoning is to avoid large fish, which is more than 2 kg of susceptible species. So this could be one of the risk management measures which the safety public health authorities can adopt in case there is a outbreak or so ask people to avoid large fish because large fish tend to accumulate a lot of these toxins because this is a cumulative effect because the toxin comes from the algae. So the bigger fish consume more of these algae and they have more toxin in their body. So this is how public health control is exercised in some of the regions. So this is what I mentioned. FAO and WHO got interested in this, and particularly since there are no codex related toxins. So to summarize, what is the state of knowledge that we have today on the Sigmoidal fish poisoning, the toxins, and what are the public health measures people can adopt in different countries to manage fish safety. So now I will touch upon another bacterial pathogen which has also shown impacts due to climate change. Now, Vibrio species. Now, of course, we are all familiar with Vibrio cholera, which causes the disease cholera. But there are a number of other Vibrio species, but most of them are not pathogenic. There are only 3 major species, Vibrio cholerae, Paralytic, and Volnificus, which are important human pathogens. But again, among these, there are environmental strains which are not pathogenic. Even in Vibrio cholerae, there are over 200 serotypes, and only the serotype O1 and O139. So we generally, if you send a sample of stool to the laboratory, they will identify, okay, Vibrio cholera, O1, Ogawa, or Inaba isolated. So this serotype thing is done. And so far, only 2 serotypes are known to produce the disease cholera, because we need to differentiate the disease cholera from other diarrheal diseases. See, other Vibrio cholerae may cause a mild gastroenteritis, which is different from cholera, which is characterized by a massive loss of fluid from the body due to severe episodes of diarrhea. So we need to differentiate this. Then there are other bacteria like Vibrio parahemorticus. And Vibrio parahemorticus is another foodborne, fishborne pathogen. And among these, only 1 to 2% of environmental strains are actually human pathogens. They possess virulence factors that cause disease in humans. So this summarizes the source of some of these Vibrios which cause human disease. So they could be through shrimp or fish or mollusks. So they can be present associated with the plankton, the phytoplankton, zooplankton, particularly zooplankton, which contain chitin. So these could be one of the sources of these type of Vibrios. So I will talk about Vibrio parahemorticus because there is interesting evidence to show that this has migrated across continents through the impact of ocean currents. Now, Vibrio parahemorticus is common in estuarine and coastal environments. There is global occurrence and associated with all types of animals in brackish water, zooplankton, mollusks, crustacean, and finfish. And the ecology is influenced by temperature and salinity. And in tropical environments like ours, it is detected throughout the year. So when we did studies in Bangalore, we could find them throughout the year. So the conditions are suitable. But in temperate climate, these are detected only in summer months. And in winter months, this undergoes overwintering in the sediment. So it can be detected in the sediment, but not in the shellfish. So till the 1990s, this organism was considered only tropical. In fact, in international fish trade, there were even rejections. So if they find Vibrio Species, whether it is environmental or not, they used to reject the fish, saying that, oh, your fish is containing this dangerous pathogen. Though, of course, at that time, there was not much knowledge on the pathogenicity of this Vibrio Species. But then, in 2004, there was an outbreak of gastroenteritis in Alaska, which is a very cold region. And so here it was due to a local shellfish harvested from Alaska, which was involved in this gastroenteritis. And so this changed the thinking on the distribution of this organ. And this outbreak in Alaska is about 1,000 kilometers north of places from where any previous incidents of Vibrio Species were reported before. So this again shows the change in the geographical range through which these type of pathogens are occurring. So why the outbreak occurred in Alaska in 2004 is because the temperatures were higher than 15 degrees. So Alaska, normally the temperatures are much less, below 10 degrees, but at that time there was extreme high temperature, again an impact of climate change. So the temperatures rose above 15, and this led to this outbreak of Vibrio Species in Alaska. And subsequently, people have been looking at Vibrio Species and its association with the zooplankton, which contain chitin. Now, the chlorophyll, which is present in the phytoplankton, may also influence the ecology by influencing the ecology of the zooplankton. And number of studies have been conducted also in Europe. And the study done in Spain, the Galician region of Spain, indicates that Vibrio Species is found even in offshore areas. So earlier it was confined to mostly nearshore areas. So this study showed that even in offshore areas, which are far from the coast, they could find Vibrio Species in association with zooplankton. So this shows that the ocean could be carrying this pathogen. And genetically similar organisms were found in zooplankton in estuaries and offshore regions, spread over 1,500 kilometers, suggesting a role for zooplankton in the spread of this organism. Now, once it is found in the oceanic waters, there can be different ways in which it can spread. We are all aware that there is a lot of traffic, the vessels, ships which are going across the sea, and many times, particularly the oil tankers, because in particularly countries like India, of course, we don't produce enough of our crude oil. So we import crude from other countries which produce crude. So we refine this oil and then export refined oil. Now, when the crude comes to India and when the tanker has to go back, now these tankers have to be filled with water. So they have to be filled with seawater. And then when they go to the next port, when they have to fill in oil, so they have to discharge this water. So this type of water, which is used for balancing the ship, is called ballast water. Now this ballast water is now suspected to carry many of the pathogens. So now there are already international regulations on what should be the quality, where you should discharge this ballast water, and will it spread pathogens which will affect human health in the coastal regions. So now this study in the Galician region of Spain showed that this Vibrio Species can be found in offshore waters, so it may be present even in the seawater. Now there are different serotypes of this Vibrio Species, and one serotype is of particular interest because in 1996, in India, studies done in India in the National Institute of Cholera and Enteric Diseases showed that most of the outbreaks that occurred in India at that time was due to this one variety, O3K6. And then they found that this variety has spread to other parts. So they detected this variety in Peru and also in Chile. So how did this serotype, which was detected in India, could spread to the South American continent. And large number of cases were reported again in 2003 to 2004. And in Chile, over 10,000 people were affected by this strain because shellfish is a very important commodity that is produced in Chile. So through this shellfish, there were a large number of people who became sick. So how did this spread occur? So this illustrates the global spread of what we call pandemic. Of course, this is not actually a pandemic in the real sense of the term, because when you say pandemic, then like we see corona today, large number of cases in different parts of the world. Now here the term pandemic was used only because the same strain was found in different continents. So it spread globally. So the term pandemic was used, though of course this is not an appropriate term and it's not a WHO used term. But in literature, people have been using the term pandemic strain of Vibrio Species. So first detected in India in 1996, and then parts of Southeast Asia, and then in South America. And also now later it has also been detected in North America. So how did this global spread of this strain occur? So people have been tracing the weather events that have occurred during this period, and these studies and molecular studies on the strains which were involved in these outbreaks showed that El Nino episode provided the corridor for displacement of Asian pandemic Vibrio Species strains to the American continent. So El Nino is becoming again a weather-related event, and it is characterized by the arrival of equatorial warm waters in a sequence of waves. So in 1997, El Nino affected South American coast for about 6 months. So repeated invasion by these warm water currents from the equatorial warm waters occurred in into the coast of the South American coast. So it has been suggested that recurrent invasion of tropical masses of water might have resulted in repetitive sources of contamination of these Vibrio Species, which became then established in the South American coast. So this is this was hypothesized at that time, and see 1995 actually later archival strains studied in 1995 showed that the strains which were first detected in India in 1996 were already present in some countries in Southeast Asia, and then it has spread to South America in 97 due to this El Nino event. So what is the evidence that we have? So there is data on sea surface temperatures and anomalies in the ocean currents that have taken place during this period and El Nino. So this spread occurred during these events when there were El Nino and increase in the temperature. So here you can see the illustration of this case of Vibrio Species in 1997, and there was also spread of Vibrio cholerae in Peru in 1990. So this has also been now traced to the invasion of warm water currents coming through these El Nino events. So here you can see these are the warm water currents coming to the South American coast. And then if you look at the cases, so early cases appeared in this region. And as the warm water current expanded southward, the cases also extended southward and the cases expanded. So you could find clear relation between these ocean current, warm water current, and the disease events that were taking place in the South America caused by this organism. So what I would like to summarize is that events related to climate change have caused increased incidence in algal blooms, fish and shellfish toxicities, and the geographical range of certain harmful algae is expanding. And events like Sigmoidal fish poisoning is occurring now in regions which were not known to be endemic. So it's spreading to new geographical regions. And events like El Nino have caused transoceanic spread of bacterial pathogens like Vibrio cholerae, which arrived in Peru in 1990, and Vibrio paramolyticus, which arrived in Peru and Chile in 1997. And there are also a number of other pathogens. Of course, I want to go into details of this, but a few cases that I mentioned here illustrate the importance of climate change and the impact on health, particularly through the ecological disturbances that are being caused in the oceans and increase in the toxic species. So even in the terrestrial environment, we will definitely find changes like this, increase in the vector distribution and more vector-borne diseases and more zoonosis is other. So we need to be extremely careful about this and see what we can do to minimize the impact of climate change. Thank you very much.
Thank you very much, sir, for such an informative lecture. Now, I would like to propose the vote of thanks. I would like to thank Nitte deemed to be University for providing me giving us the opportunity to organize the program and providing all the necessary facilities required to organize the program. I would like to thank the Vice Chancellor and Registrar Ma'am for giving us permission to conduct the program. I would like to thank Professor Dr. Muditaya sir who had agreed to give the opening remarks, but due to last minute commitment, he could not give the opening remarks. I would like to thank our Dean, Professor Dr. U.S. Krishnanayak sir, for all his support for organizing the program. I would like to, my sincere thanks to Professor Dr. Smita Hegde ma'am, who has always supported us and encouraged us to organize the program. I would like to thank all the participants who have registered for the program. I would like to thank the staff developmental cell and the IT department for helping me to conduct the program. Thank you everyone for the patient hearing. The feedback link has been placed. Kindly fill in the feedback form to receive the e-certificates. Thank you very much, sir.